Namaste to all students. Khushbu here. Myself, JV Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about transcription, and this session is related to molecular biology course of program MSc Microbiology and MSc Biotechnology second sem. So, first of all, what do you understand by the terminology of DNA transcription? Means the formation of RNA from the DNA. This process is known as basically DNA transcription. If we talk about the flow of the genetic information, so first of all, DNA transcribe and they can prepare the RNA. Major part of RNA is messenger RNA. Now, this process means transcription process occurs basically in the nucleus portion. After the preparation and after the production of messenger RNA, it can transfer from the nucleus to the cytoplasm where it produces the proteins by the translation process. Means there are three different terms or three different processes. First is replication, in which DNA prepares new DNA in the nucleus, and second one is the transcription, where DNA prepares and produces the RNA in the nucleus, and third process is translation, which occurs by the RNA and they prepare the protein molecules in the cytoplasm portion. So the genetic information flows basically from the DNA to messenger RNA and then to the protein synthesizing machinery. So this is the basic flow of the genetic information. The synthesis of an RNA molecule from DNA is called replication, as I have already told you. So all eukaryotic cells have 5 major classes of RNA. First one is the ribosomal RNA, rRNA. Second is messenger RNA, means mRNA. Third one is the transfer RNA, means tRNA. Next, fourth one is the small nuclear RNA, and final 5 is the microRNA, miRNA. The first three, ribosomal RNA, messenger RNA, tRNA, are involved in the protein synthesis, while the snRNA, small nuclear RNA, are involved in the messenger RNA splicing, means RNA processing, and the regulation of the gene expression. If we talk about the similarities in between replication and transcription process, so the processes of the DNA and RNA synthesis are similar in that they involve the general steps of the initiation, elongation, and termination with the 5' to 3' polarity. Means the newly synthesized strand will move towards the third dash end from 5' means from the 5' end to the third dash end, this will be the direction. Second similarity is both are large multi-component initiation protein complexes are involved in the process, and the adherence to Watson-Crick base pairing rule. Both processes are basically adhere to the Watson-Crick base pairing rules. Now we can discuss about the differences in between replication and transcription process. So ribonucleotides are used in the RNA synthesis rather than deoxyribonucleotides. Deoxyribonucleotides are used in the DNA replication process. On the second difference is uracil replaces thymine as the complementary base pair for adenine in RNA. Means if DNA is having adenine at any specific point, so in the process of synthesis of RNA, complementary sequence will be uracil, not thymine, because it is RNA and in the RNA strand there is no thymine. Uracil will be present in the place of thymine. So that's why when the DNA transcription process occurs, so uracil comes in the complementary pairing with the A and adenine, which is already present in the DNA. So uracil comes and binds to the previously chain in the RNA. And the third difference is a primer is not involved in the RNA synthesis, involved in the DNA synthesis, DNA replication process. Fourth difference is only a portion of genome is transcribed or copied into the RNA. Means if we require a specific portion of DNA, like we want any specific region RNA, so only that region will be transcribed, not the whole genome, where the entire genome must be copied during the DNA replication. Means in the DNA replication process, complete genome will be duplicated, but in the case of transcription process, we in which portion we require for the transcription, only that portion will be transcribed, not the whole genome. Fifth difference is that there is no proofreading function during the RNA transcription, but in the replication process, proofreading basically occurs. So these are the basic 5 differences in between replication and transcription process. Now we can discuss about the template strand used in basically transcription process. So the strand that is transcribed or copied into an RNA molecule is referred to as the template strand of the DNA. The other DNA strand, which is non-template strand, is frequently referred to as the coding strand of the gene. Here you can see the diagrammatic representation, where you can simply understand more about the template strand. Strand of DNA, which is basically used in the synthesis of messenger RNA, is known as template strand, and the second strand of the DNA, which is not involved in the RNA process, RNA transcription process, is known as coding strand. This strand is as a template strand because it is basically used by the transcription machinery for the synthesis of messenger RNA transcript. So that's why we call it template strand. The information in the template strand is read out in the 5' to 3' direction. Means DNA template strand basically 3' to 5' direction, and it is having all the information, and the sequence of the ribonucleotides in the RNA molecule is complementary to the sequence of deoxyribonucleotides in template strand of the double-stranded DNA molecule. In the coding strand, which is complementary strand, the sequence is same as that of the sequence of nucleotides in the primary transcript. We can understand this by diagrammatic representation more. You can simply see this is the DNA, which is template strand. It is basically used for the transcription process, and all the sequences are complementary with the messenger RNA molecule, and this strand, which is used in the transcription process, is known as template strand, and the opposite DNA strand is basically similar to the messenger RNA except uracil. You can simply see here, like in the template strand, if T is here, so messenger RNA will have A, complementary. If it is having A, so here is uracil. G, C, because G pair with the C. G again C, and A pair with the uracil. If we compare messenger RNA sequence with the DNA coding strand, it is almost similar, except uracil strand. Means if we see here like A, but T is in DNA coding strand, so in messenger RNA here will be uracil. C, 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 T. So again uracil, G, 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 T, then uracil, A, G, A, G, 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 A, T, again uracil, C and C, 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 T. So uracil, A, G, and G. So you can simply see that DNA coding strands are completely similar to the messenger RNA which has been synthesized except at the uracil point, and the DNA template strand is complementary to the messenger RNA sequence. So a transcription unit is defined as that region of the DNA that includes the signal of for transcription initiation, elongation, and termination process. Here we use DNA depend. Here we use DNA dependent RNA polymerase in the complete transcription process. Cells use this kind of a RNA polymerase enzyme which is DNA dependent, and it is responsible for the polymerization of ribonucleotides into a sequence complementary to the template strand of the gene of the DNA. The enzyme attached at the specific site of the DNA. This specific site is known as promoter because it is basically the initiation point or promoter point from where when this RNA polymerase attached to the promoter region, so transcription process starts or initiates. This is followed by the initiation of the RNA synthesis at the starting point, and the process continues until a termination sequence is reached. Here again, we can simply understand what is the promoter region and how it looks like. So you can simply see this is complete transcription unit, means this region of the DNA will be transcribed and will create the RNA region. So the portion of DNA, this portion of the DNA is known as transcription unit. It is having promoter region, and this is transcribed region of the DNA. In the promoter region, you can simply see -35 region, -10 region, and this one is the transcription start site from where transcription process starts, and this is coding region, and this is completely transcribed region, and this is the termination signal which gives the signal to terminate the transcription process, and these are the promoter region. Now, first of all, you have to discuss about the primary transcript. So, RNA product which is synthesized in the 5' to 3' direction is the primary transcript. In prokaryotes, this can represent the product of several contiguous genes. In mammalian cell, it usually represents the product of a single gene. The 5' terminal of the primary RNA transcript and the mature cytoplasmic RNA are identical. The starting point of the transcription corresponds to the 5' nucleotide of the messenger RNA. This is designated position +1 as the corresponding nucleotide in the DNA. The number increases as the sequence proceeds downstream. The nucleotide in the promoter adjacent to the transcription initiation site is designated -1. These negative numbers increases as the sequence proceeds upstream, away from the initiation site. This provides a conventional way of defining the location of regulatory elements in the promoter. The upstream and downstream method you can understand by this slide. Here you can see this is the promoter region of transcription unit, and this is the start point, and this complete RNA coding region, or you can say transcription site or region. This is the terminator region, and here termination transcription termination site, and this is the transcription start site. This is completely DNA strand, and in the left side it is upstream, in the right side it is downstream. So you can simply understand about the concept of the upstream and the downstream. If we talk about the bacterial DNA dependent RNA polymerase, so the DNA dependent RNA polymerase, which is RNAP of the bacterium E. coli, exists in approximately 400 kilodalton core complex consisting of two identical alpha subunits, similar but not identical beta and beta dash subunits, and the omega subunit and a sigma subunit. Beta is thought to be catalytic subunit. RNA-dependent RNA polymerase, a metalloenzyme, also contains two zinc molecules. The core RNA polymerase associates with a specific protein factor, which is sigma factor, that helps the core enzyme recognize and bind to the specific deoxynucleotide sequence of the promoter region to form the pre-initiation complex, which is PIC. Bacteria contain multiple factors, each of which acts as a regulatory protein. Now we can discuss about the mammalian DNA-dependent RNA polymerase. So, mammalian cell causes three distinct nuclear DNA-dependent RNA polymerase. RNA polymerase 1, which is required for the synthesis of ribosomal RNA. RNA polymerase 2, which is required for the synthesis of messenger RNA and miRNA. And RNA polymerase 3 is basically required for the synthesis of tRNA, snRNA, and 5sRNA. Means one ribosomal RNA, 5sRNA, synthesized by the RNA polymerase 3, but rest of the ribosomal RNA are synthesized by the RNA polymerase 1. If we talk about the steps of RNA synthesis, so it basically involves initiation, elongation, and the termination process. Now we can discuss about the overview of the transcription process by this diagrammatic representation. Here you can simply see the transcription unit of the DNA, means the region or the portion of the DNA which is basically going to transcribe and basically involved in the production of the RNA. So that active region of the DNA is known as the transcription unit. So transcription unit is having promoter region, and it is also having a terminator region and start point. So first of all, binding of the RNA polymerase and the local DNA unwinds. This RNA polymerase comes and it binds to the promoter region and unwound the DNA molecules. Now antiphase comes and initiation of the RNA synthesis occurs. This RNA strands start to synthesize, and further third step, which is elongation of the RNA, happens, and more antiphase are start to bind, and further elongation process occurs. But at the point where terminator region is present, it gives the termination signal to the machinery of transcription process. So the transcription process basically terminates, and the RNA which has been synthesized, it basically completely separated from the DNA region. So, but one more thing, at one time only one strand will be active for the transcription process. Not both strands will involve in the transcription process at the same time. So only one RNA strand synthesized at the one time. So this is the overview of transcription process. And if we talk about the transcription cycle, so transcription cycle is basically involves all these steps. So you can again see in this termination cycle. So transcription cycle, first of all, template binding and the closed complex formation, and then open complex formation, ATP and NTP comes and chain initiation occurs, and then NTPs, more NTPs, and this promoter clearance occurs, and then more NTPs, and finally chain elongation occurs, and then chain termination and RNA polymerase releases. And that's how RNA start to synthesize during the transcription cycle. If we talk about the initiation of the transcription process, so initiation of the transcription basically involves the binding of the RNA polymerase holoenzyme to the promoter region on the DNA to form pre-initiation complex. Means, first of all, you have to know about the transcription initiation enzyme complex. So basically, it is having alpha 2 identical units and beta 1, beta and beta dash 2 units. But this enzyme is known as core enzyme. When sigma factor comes and binds to this core enzyme, so it creates a holoenzyme and now it is active. So it will come and it will bind to the promoter region of the DNA to form a pre-initiation complex, which is known as PIC. And the characteristics consensus nucleotide sequences of the prokaryotic promoter region are highly conserved. So this is the basic overview of the transcription process in this session, and the detail of each step of transcription process we are going to understand in our next session. So for this session, there are some important key points like how many RNA polymerase are involved in the eukaryotic transcription, and in prokaryotic, how many RNA polymerase are involved. As we know, in the bacterial cell or in the prokaryotic cells, only one type of RNA polymerase is enough. But in case of a eukaryotic cell, there are three types of RNA polymerase: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. All these three categories are involved in the different type of RNA synthesis, and this is major important key point of this transcription process. One more thing, examiner also can ask about the basic difference in between the core enzyme and holoenzyme. What is the composition of the core enzyme and what is pre-initiation complex? We can examiner can also ask about the full form of the PIC. So you have to know about that, and what is the basic difference in between coding region, coding strand, and template strand of DNA during the transcription process. And one more thing, examiner also can ask about the 5 basic differences in between transcription and translation process, transcription and replication process. So that's how this session is sum up, and this session is powered by digital version 2.0 JAYOTI VIDYAPEETH Women's University